So welcome to the class, students. Today we are going to study the resequencing. So this is resequencing is helpful in the identification of SNPs variation and in the detection of genetic variation. So what is resequencing? First, we will discuss what is resequencing. Resequencing is the process of determining sequence of a gene or other sequence known to be related to a reference sequence. So, as the name implies, you can see resequencing means sequencing we are doing again. Okay. So, what is the need to do the repeat the process? The process of because we want to determine the what may be the differences if we compare the sequence that we are sequencing now. We can compare that sequence with the reference sequence that we already have had from the previous methods. From the previously we have the sequence, we have the reference sequence. Now again we are doing the sequencing. Then we can compare. Okay. By the comparison, we will come to know what may be the difference. If there is any mutation that can be identified, if there is any SNP that can be identified. So this is also useful in case of disease abnormal person. The genome of normal person can be taken as a reference and genome of abnormal person or gene of abnormal. So that can be compared to that. So resequencing is always helpful in the diagnosis of disease or in the medical areas. So in the identification of SNPs and genetic variations.
मैम
Okay. Yes.
मैम एक बार आप क्या हिंदी में समझा सकती हैं जो भी आप पढ़ा
हिंदी
रही
में
हैं? मैम एक बार इंग्लिश में एक बार हिंदी में समझा दीजिए तो ज्यादा अच्छे से समझ में आ जाएगा.
I will provide you the recording of these lectures so you can listen it. If you will listen it again and again, so you will understand what I am saying. Okay.
बट मैम एक्चुअली में यूपी बोर्ड्स के हैं तो इसलिए थोड़ा सा प्रॉब्लम होता है. तो आप एक बार हिंदी में और एक बार इंग्लिश में समझा देंगे तो ज्यादा अच्छे से सबको समझ में आ जाएगा.
Okay, you have to write down in your copies in English only, but you have to write down that. Okay.
यस मैम बट समझने के लिए तो मैम
Okay, I will provide you that
समझ
next
नहीं आता
time.
मैम ज्यादा.
That is why I am going very slow. I'm not taking lecture very fast. Okay. So I
यस
will
मैम.
provide you the YouTube link of this video also. So resequencing is do it again. This is not a difficult sentence. Resequencing means हम उसकी sequencing कर रहे हैं. What is our interest? कि कोई single nucleotide polymorphism है उसको हमें identify करना है. कोई भी वेरिएशन है हम उसको आइडेंटिफाई करना चाहते हैं. वी वांट टू नो अबाउट दैट कि means what may be the manifestation of this sequence. So on the level of the sequence, we want to identify what are the nucleotide change. Okay, so that is the determining the sequence of a gene. What is determination? That means we are doing empirically, in the practically, experimentally, identifying that. Prediction we do in the bioinformatics. We use the prediction word when we do it in the computer, but this is now we are doing it experimentally. If we are doing this, that is the process of determination. So always in the comparison we have the two. One is the reference sequence and one is the unknown sequence that can be compared to this reference sequence. ठीक है. एक वो चीज होती है ना जैसे हम पहले से जानते हैं. If you will see any new thing in the environment also in your surrounding, then you will ask from anybody else कि what is this, or you will try to identify that things. कोई भी नई चीज आप देखते हैं आप आइडेंटिफाई करने की कोशिश करते हैं कि इसका क्या यूज हो सकता है, what may be the application. तो आप क्या करते हैं, आपके अंदर जो भी सेव है ब्रेन में उससे कंपैरिजन करते हैं. You do the comparison because reference information is saved within your brain. You may have information that you have already seen the things in the nature or in your environment, but now today if you you are observing some new thing, you are in the environment, then you can see that this is a new thing or this is similar to that, कि वो किससे मैच करती है, उसके बेसिस पे आप उसकी पहचान करते हैं कि इसकी एप्लीकेशन, इसका काम क्या हो सकता है और ये दिखने में कैसा है। तो जो भी हमारे पास पहले से इंफॉर्मेशन होती है, प्रायर इंफॉर्मेशन जो होती है, उसे हम रेफरेंस कहते हैं। whatever information we have already, that is called reference। रेफरेंस है वो पहले से। अब कोई नई चीज आई है तो हम उसको कैसे आइडेंटिफाई करते हैं, जो पुराने उसके बेसिस पे कि इसका ये फंक्शन हो सकता है। Okay, so this process is carried out to look for mutations that have not been previously characterized। मीन्स पहले से वो म्यूटेशन नहीं आइडेंटिफाई हुई है। सो इन दिस प्रोसेस वी मे आइडेंटिफाई द न्यू टाइप ऑफ म्यूटेशन। सो द resequencing is basically done to detect the mutation। So this is very helpful in case of mutation and SNP। That's why we are discussing this here। Okay, genome sequencing and genetic variations। So what are the different, means there are new advancement in the technology। Now we will discuss some new advancement, means what are the technology, something about the introduction of the technology। Technologies for selecting segments of large genome for resequencing। So we need to select segment, means where we want to detect the mutation। So resequencing will reveal biologically important sequence variation। Means what is biologically important, means it has the function, it is involved in the biological process। Means the mutation is a part of that mutation, that mutation has role in the biological process, so we have interest in that। So but we need to select the segment of the larger genome। There are so many larger genome, we need to select the segment where the mutations, where is the possibility of mutation। Now that the reference genome sequences for many organs are available in the databases, you can see, you can take them as a reference sequence, reference genome sequences, because that are available in the databases। Sequences of the model organisms are also taken as the reference genome sequencing। Cataloging sequence variation means उनका आइडेंटिफिकेशन करना, उनका नाम रखना, उसे हम कहते हैं कैटलॉगिंग। कोई नंबर होता है, लाइक प्रोटीन हैज द एक्सेशन नंबर, जीन हैज द जीन आईडी। In that way, sequence variation also has the cataloging number। Okay, so variation have been also provided this type of identification mark or name, so that we can easily identify, we can call them by their name। And understanding its biological consequences has become a major research in। So we want to understand what may be the biological consequences of this mutation or these mutations। So reference genomes are available। I'm telling you the name of one database that is called Online Mendelian Inheritance in Man, OMIM, Online Mendelian Inheritance and in Man। So this is one of the database that provide you the data for the human genome that are hereditary। But if you want to see the changes, variation, then you need to search in the DB-SNP database on single nucleotide polymorphism। So in the OMIM, we can have some details of the genetic disorders also that is associated with the genes, and those mutations are hereditary। In the genetic disorders, there are certain genetic variations that are hereditary। However, for large eukaryotic genomes, such as the human, this is the large, means human has a large genome। So high throughput sequencing technology allow deep sequence-wide sequence coverage of a small number of individuals। So, but if we want to sequence the whole genome, so but the small number of the individuals can be sequenced। because it is very difficult process. As in the NGS also, and in the previous classes we have discussed about how the genome sequencing of the larger genome is difficult process. We have to cut down. Cut down the larger genome into multiple fragments. Then we do the sequencing of those fragments individually. So this becomes a very difficult process. This is not feasible actually. Realistically, it is not possible to sequence, uh, to take the genome of from all the humans and to sequence them. This is not an easy process. But we are moving forward in this process. Now we are able to sequence. But we are doing the development in the technology. High throughput means we want to sequence many more genome at the same time. Resequencing the genome of many individuals for which there is reference genome allow. Why we use the reference genome? This allow investigation of relationship. Because by using the reference genome, we can compare the test genome which we are testing now. Investigation of relationship between sequence variation, normal or disease phenotype. Means what are the variation between normal and abnormal. So we can compare the sequences, we can compare the genome. Especially we should compare the genetic sequence variation of normal and abnormal. Okay, so then we can identify the causes of the disease. Some of these platforms permit 100-fold greater rates of DNA sequencing. Some of these platforms permit 100-fold greater rates of DNA sequencing. Now we have the platform, some of the advanced platforms that permit the 100-fold greater rates. Means sequences 100-fold increase. रेट जो है सीक्वेंसिंग जितना हम पहले करते थे उससे 100 गुना ज्यादा अभी हम कर रहे हैं सीक्वेंसिंग एट अप्रोक्सीमेटली 100-fold लोअर कॉस्ट पर बेस. और जो कॉस्ट है वो रिड्यूस हो गई है. नाउ कॉस्ट हैज बीन रिड्यूस मींस 100 गुना कम हो गई है वो कॉस्ट अब पहले जो कॉस्ट थी उसके कंपैरिजन में. So this has become cheaper now and rate has been increased. If this new sequencing power could be targeted to limited areas of larger genome, means if we select the segment, selection of the segment will be useful. We should also means we should focus on the small segment where the mutation exists in the larger genome. So this will become more useful in that case. कि अगर हम पूरे जीनोम पे ना फोकस करके जहां म्यूटेशन है उसको हम सीक्वेंस कर लें, it would become feasible to study variation in these regions in thousands of individuals. तब हम क्या कर सकते हैं मीन्स कि पूरे जीनोम को ना सीक्वेंस करके इस केस में क्या किया है कि एक सेगमेंट हमने ले लिया जहां पे म्यूटेशन है, पॉसिबिलिटी है, we have taken out that segment and we have done resequencing for that. Then we have compared that sequences of those regions with other individuals, sequences of the other individuals. Means specific regions we are taking from the genomes. So this will become feasible. This will become an easy process. It will be cheaper process also. So right now we are in the condition that we can resequence the segments of genome. क्योंकि एक बार सीक्वेंसिंग करना ही बहुत डिफिकल्ट होता है, फिर उसमें बार बार रीसीक्वेंसिंग करना, so that is a difficult process. So and costly process. So what we can do, we can take the segment, we can take the limited areas of larger genome and we can sequence and we can do the comparison in the thousands of individuals. Then only it is possible to study the thousands of individuals. This configuration of the new sequencing technology would allow the direct and practical application. So in this way, this will allow the direct and practical application to studies of human disease. Means what are the causes, what are the phenotypic appearance, means how the SNP and DNA polymerase is responsible for the manifestation of different symptoms of disease. So at the phenotype level, we will able to observe. So we can compare the sequences variation with the human disease. This is a process in the NGS data analysis. We have also discussed about how the different with the advancement in the technology in the next generation sequencing data provide the more information from all the field. It integrate information data from all the fields that are related to sequencing. So that is the new generation sequencing platform. So within the NGS, we have studied about the different platforms, how the statistical analysis is involved there. And there are the different platform where we can do that. Different platforms are connected with each other to perform the different kind of analysis. So NGS, in the NGS data analysis, we have the integrated technology. So that give you a small number of complete genome screen for variation. So we have already discussed about this in the NGS data analysis, small number of complete genome screen for variation. So here the small number means we have taken the few means individuals from the complete genome. We can compare the segments also, where we can find out where is the possibility of the variation. And this is the enrichment process. There are the different procedure, there are the different technology, new technology also that involved in this process, and combined methods are used in the next generation sequencing. Here you can see the fragments. So this is the new generation sequencing platform that provide us the larger number of genome screen for variation in enriched component. So previously we were able to sequence the smaller number of complete genomes. So now we have so too much technology. We have different technology that we can combine together. So due to that, we are able to sequence large number of genomes, and we can screen for those genomes for the variation. We can identify the variation by doing comparison with the reference genome, because databases are also connected with them. The NCBI databases and other databases are all connected on the same platform. So now we have this kind of platforms that we can do. Miss Wet Lab information is integrated, have been analyzed within the computational network. Okay, then it is compared with the databases, the information that is stored in the databases. So where we do the screening. So on the basis of that, we can identify the mutations and variations. So how this happens? This is the comparison of the new genome with the reference genomes. So there are the 4 new studies in the nature method. I'm teaching you something new. That I have taken from the website. There are the 4 reports in the nature method in nature genetics. This is the name of journals of nature that describe the novel strategy that are very new for processing whole genomic DNA that result in substantial enrichment. So that is up to 2% of the genome. So we are doing effort. Now we are in the developing phase, in the development process we are doing. So coupling of these enrichment approaches is here. Coupling means we have combined the different approaches to give the best result. So this is what we call the enrichment approaches to high throughput sequencing technologies. What is means high throughput means we can analyze thousands of genes or more number of genomes simultaneously. High throughput means input is more here. You can give more input here. Promises to deliver a huge body of information that can give you the huge body of information about DNA sequence variation in population. So here we are talking about population. That means it is on the large level. If we couple those methods together, okay, to the high throughput technology, sequencing technology, then we can have the huge body of information about the variation in population. We can detect the changes and variation in the population. But that is only possible by the combination of the technology, coupling of this. So what is this enrichment? We have the multiple methods there. We have the information from multiple platforms. So here a new phase in the genetics of human and other organisms with larger genome. Means now we are in the condition that we can do it. We can do the comparison. We can, because we integrate the information from different fields in different databases. On the basis of that, we are screening the gene mutations, screening the variation, genetic variation. We are identify the variation that exist within the population. Okay, so this was all about, you don't need to go into the details. If you want to know, you can search on the, I can give you the material regarding this. Because this is, resequencing is useful in the identification of genetic variations. So that's why we are discussing this. In the NGS data analysis, we have discussed so many things there. Okay. So about those technology, we have what previously exist, we have already discussed about those methods. And there are something new reports. So I have mentioned here. So in this article, the reference has been given, this third reference. So in this article, you can have the details of those. Reference of that. Okay. Short tandem repeat, short tandem. Okay. STR. What is this? Short tandem repeats means tandem is that is not random, means this is highly organized. In the random that is not organized, but in the tandem repeats, tandem means that is as antonym of the random. So tandem is highly organized. Short tandem repeats. Short tandem repeats, STR are sequences comprising a number of repeats of short 2 to 5 nucleotide subsequences. Means like 2 to 5 nucleotides of repeat subsequences that is found thousands of times, hundreds of times. So there are the different types of repeat, generally thousands of times, 1,000 repeats, 100 repeats. So there are the different pattern. There is some pattern in the repetition. Within the same sequence, we have the repetition of the 2 to 5 nucleotide subsequences. Typically, such repeats are scattered throughout the genome. So these are called short tandem repeats. And are flanked by unique sequences. Unique means that is not found else. That can be used to target polymerase chain reaction. Means in the polymerase chain reaction amplification, we can identify those. We can use those sequences in the identification of gene also. In the identification of the locus of gene. अगर हमें कोई gene identify करना है तो उन sequences को हम use करते हैं as a marker. These are also used as a marker in gene mapping or in the identification of the gene locus also. So most STR are available in copy numbers, means copy होती है ना, 2 nucleotides है, then 2 nucleotide repeat and 5 nucleotide repeats occur for hundreds of times, for thousands of times. So depend on that. It depends on the copy number. So making them valuable genetic markers. So short tandem repeats means 2 to 5 nucleotide subsequences. There is a repetition of 2 nucleotides and 5 nucleotides. So see, and others type of repeats are microsatellite and mini satellite. So in that we can have the large number of nucleotides, means the greater than the short tandem repeats, STR, greater than the short STR. Short STR means short tandem repeats like 2 to 5, but in the micro satellite we can have many more also. In the mini satellite also we can have the more number of nucleotides that are repeated. Short sequence length polymorphism. So these are all kind of the STR, microsatellite and mini satellite. So these are the subset of variable number of tandem repeats, which also include mini satellites. Sizes of the mini satellite repeats are that is more than the 100 base pair repeats. Okay, the repetition pattern is more. So variable number of tandem repeats, VNTR, variable number of tandem repeats. So variable number of tandem repeats are used in the identification of ancestry, like father-son relationship. So these are very helpful in the DNA fingerprinting. These markers are helpful in the identification of the genome, means in the detection of relationship phylogeny. So these are exploited in the DNA fingerprinting method. Okay, so these are the references that I have taken this material from these papers. You can read them. If you have any question now, you can ask. There's so many data that is available. You can search about the nodes on the short tandem repeats tandem. I can provide you more information on this. So these are helpful in the gene mapping. We have already discussed about this also, I think in the previous lectures. I have covered also this. In the molecular markers, in the crop improvement. So these are very, in the crop improvement also we have discussed about this. Do you have any question now? If you have any question, you can ask.
Ma'am, what is the work of next generation sequencing?
Work?
हाँ मैम.
Work of next generation sequencing means that, as we have already discussed in the class, we have so many multiple methods that have been combined there. You just go through those slides. In that we have the flow charts have been given. So how they are combining the different. What is the enrichment process? Means we are using multiple methods, and we are gathering information from all the platform. Then we are screening. So these are helpful in the screening of the variation. These are helpful in the medical field. Means we are able to identify the genetic variations that is responsible for the disease. So in that way it is very helpful. We can work on those disease, we can correct them. So this will be helpful in the development of therapeutics or other treatment technology that can be useful. So for the screening, the NGS data can be used. So NGS sequencing is useful in that. Next generation sequencing, this platform provide us that enable us to screen the genomes and genetic variations. Because we are combining, that is the important coupling of the technology. We are combining the different methods. Then we are doing the screening. So the lot of information that is we are gathering from, there are the multiple step, that is the multiple step procedure. And we are also doing the statistical analysis and computational analysis, that everything have been provided on the platform. So there are certain different technology that is combined together, and there are different databases, information resources that are combined together, so that information from the different fields can be used in the screening of the genome. Because now today we are discussing about the genetic variation, so this is very helpful in case of disease or in medical field. And this is also very helpful in case of plant also when we talk about the crop improvement.
मैम ये फर्स्ट जनरेशन सीक्वेंसिंग सेकंड जनरेशन सीक्वेंसिंग क्या होती है?
So first generation sequencing means when we got the genome, means that is the advancement in the technology. In terms of technology, you have to distinguish. Okay, previously we had the sequences of the genomes that is available on the NCBI databases. because it is very difficult to sequence the larger genomes. So that's why the next generation sequencing methods have become more important, because in that we have combined the new methods. So previously we had the Sanger dideoxy method, that was also the advanced method of that time. So in the every decades we are doing development. So after 10 years we are on the next step. So we are on the continuation, we are working to improve the methods. Now in NGS what is important? There is the involvement of computers. We have databases. We can perform the databases, analysis, screening of the genomes. So screening is important.
तो मैम
First,
जो नेक्स्ट जनरेशन सीक्वेंसिंग है इसमें मैम कंप्यूटर को वो करते हैं, इन्वॉल्व करते हैं, तो इससे जो पहले सीक्वेंसिंग होती थी उसमें क्या करते थे मैम फिर?
हाँ नहीं, पहले सेंगर से पहले नहीं यूज होता था, जब आफ्टर द इन्वेंशन ऑफ सेंगर डाइडियोक्सी मेथड, फर्स्ट टाइम द
यस
कंप्यूटर
मैम।
मेथड्स आर वर यूज्ड फॉर दैट सेंगर डाइडियोक्सी। ऐसा नहीं है कि हम पहले से कंप्यूटर नहीं यूज कर रहे थे, वी वर यूजिंग दैट, मींस
तो इसमें मैम
आफ्टर
सिर्फ एडवांस हुआ है और कुछ नहीं हुआ है?
after the invention, after the development of Sanger dideoxy method, we have used the computer
यस
in
मैम।
the sequencing, यूज कर रहे थे, बट दैट वाज अ लोकल, है ना। उस टाइम पे क्या था,
यस मैम।
इंटरनेट टेक्नोलॉजी इतनी डेवलप नहीं थी ना। नाउ व्हाट इज द डिफरेंस, नाउ वी आर इंटरकनेक्टेड विद ईच अदर, मींस नाउ वी आर यूजिंग द इंफॉर्मेशन फ्रॉम ऑल द रिसोर्सेज, कि अब हम एक जगह बैठे हैं, दैट आई एम टॉकिंग टू यू, पहले ऐसा नहीं था ना।
यस मैम। मैम ये मैम हमने पढ़ा था कि उसमें ज्यादा नहीं बेस पेयर का मैम सीक्वेंसिंग कर पाते थे, जो जो पहले मेथड थे सिंगर
हाँ हाँ,
डाइडिऑक्सी मेथड में, लेकिन इसमें नेक्स्ट जनरेशन सीक्वेंसिंग में मिलियन बेस पेयर को भी सीक्वेंस कर लेते हैं।
नहीं वो तो हम कर लेते हैं वेट लैब में ही, और स्पेशल इसमें क्या है कि मींस वर्चुअल स्क्रीनिंग भी है ना, मींस वी आर कनेक्टेड विद द डिफरेंट डेटाबेसेस, इजीली we can use that information,
ओके
means
मैम।
due to the internet we are connected with each other, the different laboratories are connected with each other. At that time it was not like that, huh?
यस मैम।
उस टाइम पे हमने क्या किया था कि जैसे वो कंप्यूटर है, हमने इसको यूज कर लिया, तो उस टाइम पे क्या था कि वी वर नॉट इन द नेटवर्क, दैट वाज नॉट द ह्यूज नेटवर्क, बट नाउ वी हैव द नेटवर्क टेक्नोलॉजी ऑन द बेस, मीन उसके बेसिस पे क्या हुआ, कंप्यूटर में भी तो ये हुआ ना कि नेटवर्क आया, नेटवर्क डेटा मॉडल, रिलेशनल डेटाबेस मॉडल का जब वो हुआ, क्लाइंट सर्वर मॉडल आया कंप्यूटर में, ठीक है, कंप्यूटर साइंस, we have developed the client server model. After developing that, the different laboratories were connected with each other. We have developed the more information systems. इंफॉर्मेशन सिस्टम हमने डेवलप किए डिफरेंट, तो कनेक्शन हो गया ना, वो एक रिलेशनल डेटाबेस मॉडल का इम्प्लीमेंटेशन हुआ, क्लाइंट सर्वर मॉडल बना, और इसके थ्रू हम क्या, वी कैन कनेक्ट from this country to another country, we can connect the different laboratories also, that happens in the bioinformatics. This is the application of bioinformatics. So next generation sequencing platform involve more of the computational networks. इसमें नेटवर्किंग हो गई है, और वी कैन इजीली डिटेक्ट कि वेयर इज द प्रॉब्लम, वी कैन एनालाइज, सो ऑन द सेम प्लेटफॉर्म वी आर गेटिंग दैट. So with the minimum effort, if you will compare the in terms of efforts also, means it has been given 100 times वो cheaper हो गया है, क्या 100 फोल्ड रेट इंक्रीज हो गया है इसका, जो भी हम सीक्वेंसिंग करते हैं, दिस इज द मेन डिफरेंस, इसको आप लिख सकते हैं ये ना, 100 फोल्ड इंक्रीज हो गई है रेट सीक्वेंसिंग की, एंड द कॉस्ट हैज बिकम मीन्स 100 फोल्ड डिक्रीज्ड. ओके, सो दिस इज द मेन डिफरेंस. And now we have everything on the same platform. Means we are combining, we are integrating. What is the enrichment? Means we are taking the means different technology together, information from the different technologies. So that is the next generation sequencing platform. So there is the use of the bioinformatics platforms. In the NGS, we have the bioinformatics platforms, कंप्यूटेशनल प्लेटफॉर्म्स. सेंगर डाइडियोक्सी में भी हमने कंप्यूटर यूज किया था, लेकिन वहां पे इंटरनेट का इतना यूज नहीं था ना वो. ऐसे आप इमेजिन, जैसे ना आप कंप्यूटर पे काम करते हैं तो आप बिना इंटरनेट के भी काम करते हैं, विदाउट इंटरनेट यू डू योर वर्क. और जब इंटरनेट आ गया तो उसके बाद क्या चेंज आया, यू कैन थिंक व्हाट इज द डिफरेंस व्हेन यू डिडंट हैव एनी इंटरनेट फैसिलिटी. लेकिन अब तो हम इंटरनेट है ना हमारे पास, वी कैन कनेक्ट विद ईच अदर्स. So in that way, we are able to sequence a larger number of genome, screen for variation in enrichment process. अब जब हम 2002 में थे तो on that time also we can talk with each other. But कैसे? Yahoo मैसेंजर से वेब कैम हमें लगाना होता था, कैमरा, उसको यूज करके हम करते थे, तो 1-to-1 टॉक थी ना वो. बट नाउ आई एम टॉकिंग टू द multiple student. Now I'm talking to the whole class. So this was not possible at that time. Okay. So उस टाइम पे अगर किसी को दूर से पढ़ाना होगा तो एक ही स्टूडेंट को कोई पढ़ा पाता. वीडियो कॉन्फ्रेंसिंग ये सब होती थी, that was very costly. I mean the cost of that was high. और जो इंटरनेट की स्पीड भी वैसी नहीं थी ना उस टाइम पे. In the 2002, I was doing the AmTech. I've taken admission in that. Okay. So we used to chat using the messengers. So at that time we used that technology that was provided by the Yahoo. At that time we didn't use that Gmail. Now we also have the Yahoo Messenger. We can use that. So, but that was the platform we were using to send message from one side to another. So there is a change now. So because it provides a more facility, I mean we are able to large number of the genomes. इस तरह से अंतर है. जिस तरह से अभी हम multiple we are talking to. Now I am talking to the whole class. Okay, I used to take the class of B.Tech also. There we have the two batches. So sometimes I used to take class together. So in the normal class, it is not possible. That is a class of 60 students, but now I'm teaching, I used to teach 120 students also. Okay. So there is a technological changes that you have to compare, and how it has affected the genome sequencing methods. That, okay, you need to look. I mean, what is the difference? The main difference in the cost. And in the coverage also, I mean, now we can sequence more number of genomes. Cost has become low, and the rate of sequences has become increased by 100-fold. This is the difference. Okay. And you can discuss about the technology also. There is a different technology that has been used. Fragment assembly we do in the computer. That is the offline process, fragment assembly. But nowadays in the NGS analysis, we are using the internet technology, networks. Any other question? Any other question? You can type your attendance in the chat box. Just write down your roll number and name. Okay. So, okay. Just do it fast. Okay. Some of the students have done. Okay. Just type your attendance, type your name and roll number in the chat box so that this attendance can be recorded.
हेलो मैम।
हाँ हेलो।
मैम बिल्डिंग प्रोफाइल्स क्या है मैम? एक ये टॉपिक था मैम यूनिट 2 में।
Oh, building profile that we have also taken into the question and answer session.
तो
ऑफलाइन
अभी
मैंने
मैम हुआ नहीं है क्या मैम ये? अच्छा, हो
क्या,
चुका है ये?
नहीं मुझे नहीं पता, आपको पता नहीं है? ये तो बिफोर मी इट हैज बीन टॉट, और मैंने आपको क्लास
मैम
में इसके
मेरा
क्वेश्चन भी लिए हैं। हाँ, प्रोफाइल,
अच्छा
बिल्डिंग
मैम, शायद
प्रोफाइल पे
मेरा
मैंने,
छूट गया होगा शायद।
नहीं तो दोबारा अगेन वी हैव डिस्कस्ड इट इन द क्वेश्चन आवर सेशन in the class in the December. We have discussed about this. Somebody has asked the question about the building profiles, so then we have discussed about that. We will take it, okay, if you
तो
want.
मैम इसका मैम लेक्चर पढ़ा है क्या कोई कहीं पे मैम?
नहीं, लेक्चर मैंने ये मेरे से पहले ही कंप्लीट हो गया था, देन मिस विजयन सर was teaching you at that time, so he has covered that. If
मैम
you
इसे एक बार
want,
बता दीजिएगा मैम।
okay, I will revise that. That will
यस
be
मैम, इसमें मैम दो
taken
टॉपिक
in
हैं
the
ये,
next lecture.
बिल्डिंग प्रोफाइल्स।
Okay,
ओके
because
मैम।
this is the
थैंक यू
class of,
मैम।
this is the class of yeah, on the genetic variation. This is record. This is on the recording. That will be
यस
uploaded
मैम।
in the YouTube. So that
ओके मैम।
building profile will discuss in the next class. Okay.
या मैम, थैंक यू मैम।
So these are the classes that is focused on the genetic variation or some advancement in the technology. So this is the basic. In the next lectures, we will also discuss about the gene therapy and means what is the application. We'll discuss about what are the application of these things.
ओके मैम।
Oh. Just make sure you give your attendance. Okay. Just give your attendance. Anushka, Soumya, do it fast. Give your attendance, then you can leave the class. You will have to attend the another class also. Anamika, Ekanki, do it fast. Fives are left. Write down your full name actually, full name with the roll number. Who your roll number is 76 SK Rajput. Who is that? What's your name? Just write down your name with the roll number so that I can identify, because there are the multiple people by the same name. Okay. You do it fast. Kajal, Avinash. Who is Avinash in the class? Okay. Okay, if anybody is left, just do. Who is Avinash? Just give your attendance. Okay, I'm going to stop the recording. No attendance will be taken after that. No attendance will go on the record if I stop the recording. Just give your attendance. Who is left now? Okay, done. Class is over now. attendance is also over